Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Kerala SET Biotechnology question paper conducted in the year July 2023. This is part 2 of the discussion. If you are new to my channel, please don't forget to like, share and subscribe. I have also uploaded previous year's Kerala SET Biotechnology question papers. You can refer to my channel for the. Coming to the first question. Conditions for chilling preservation of fruits and vegetables is temperature nearer to. So it is nearer to 0 degrees Celsius and 85 to 95% humidity. Answer is option B. Smallest bacteria they are mycoplasmas. Answer is option D. A virus will contain. So virus can contain either DNA or RNA, not both. So answer is option C. Blood agar can be called it is a differential medium, and it allows to distinguish bacteria on the type of hemolysis produced. So the answer here is option A. The mechanism of action of penicillin is inhibition of peptidoglycan synthesis. Answer is option B. Phagocytosis was first described by Metchnikoff. Answer is option C. Immunoglobulin class responsible for local immunity is IgA. Secretory IgA you can find it on mucosal surfaces of humans and many other mammals. So it is responsible for local immunity. Then immunity obtained by vaccination is what type of immunity? So acquired immunity can be active immunity or passive immunity. Active immunity you develop after being exposed to an infection or after getting vaccine. And passive immunity you acquire from someone or something else. So active immunity is natural, that is antibodies made after exposure to infection, and artificial is after getting a vaccination. And passive immunity natural is antibodies transmitted from mother to child via mother's milk, and artificial is antibodies acquired from an immune serum medicine. So 67 they have asked immunity obtained by vaccination is artificial active. It is active immunity and also it is artificial. So answer is option D. Then next one. Immunoglobulin class seen in highest concentration in the serum is IgG. Answer is option A. Size of bacterium is commonly expressed in microns. Answer is option A. Example of sequestered antigens. Sequestered antigens are also called as hidden antigens, and these are certain self-antigens that are present in closed systems and are not accessible to the immune apparatus. Many self-antigens are found in hidden locations such as central nervous system, testis, and the eye, cornea, etc. And examples of sequestered antigen are myelin basic protein, sperm-associated antigens in some individuals following vasectomy, lens and corneal proteins of the eye following infection or trauma, heart muscle antigen following myocardial infarction. So they have asked example of sequestered antigen. Lens protein is an example. Answer is A. Hybridoma technology is used in the production of monoclonal antibodies. Answer is C. Gas pack is used for. So gas pack, it is a method used in the production of an anaerobic environment that is usually used to grow bacteria which fail to grow in the presence of oxygen. So gas pack is for providing anaerobic conditions. So answer here is option D, anaerobic cultivation of bacteria. Then minimum holding period required for autoclaving at 121 degrees is 15 minutes. Ochterlony procedure is based on. So Ochterlony double diffusion, immunodiffusion, it is an immunological technique used for detection, identification, quantification of antigens and antibodies. So it is based on double immunodiffusion. Answer is option B. Which of the following has an autoimmune etiology? So autoimmune disease out of the given options is rheumatoid arthritis. Answer is option D. Hat medium it is used in hybridoma technology. Previous January 23 paper also they have asked questions from hybridoma technology. Which type of vaccine is hepatitis B vaccine? It is a subunit vaccine. A subunit vaccine is a vaccine that contains purified part of the pathogen that are antigenic. Subunit vaccines can be made from disassembled viral particles or through recombinant DNA expression. When recombinant DNA expression is involved in the production of this vaccine, it is called as recombinant subunit vaccine. Now, hepatitis B virus surface antigens are used for the production of hepatitis B vaccine. So it is a subunit vaccine because only small parts of the virus has been used in the production of antigens. Then DNA vaccine, it is a type of vaccine that transfers a specific antigen coding DNA sequence into the cell of an organism as a means to induce immune response. So that is DNA vaccine. Killed whole virus vaccine, live attenuated vaccine, you know. Gram staining was originally derived by Christian Gram in 1884. Albert staining is for staining Volantune granules. The number of OH groups present in the below given fats can be expressed as dash number. Polensky, Ricket, Michel, acetyl and iodine. So Polensky value it is an indicator of how much volatile fatty acid can be extracted from fat through saponification. Then Richard Michel number is an indicator of non-fat compounds in edible fats like butter and ghee, and it helps in determining the purity of ghee and butter. Then acetyl number is a measure of the free OH groups in the substance determined by acetylation, and iodine number is used to determine the degree of unsaturation in fats, oils and waxes. So answer here is option C, that is acetyl, because it is a measure of the free OH group in the substance determined by acetylation. Then next one. State whether two statements given below are correct or wrong. Philadelphia chromosome is associated with chronic myeloid leukemia. Yes, that is correct. It is a chromosome 9 and chromosome 22 fusion. That is also correct. So both 1 and 2 are correct. Answer is option A. Identify the wrong statement. MAP kinases are serine threonine kinases. Yes. Cyclin dependent kinases are closely related to MAP kinases. Yes. ERKs and JNKs are MAP kinases. Yes. MAP kinases are activated by dephosphorylation. It is not dephosphorylation. They are activated through phosphorylation or as a result of their interaction with small GTP binding proteins in response to extracellular stimuli. So MAP kinases are activated by phosphorylation or their interaction with small GTP binding proteins. Then which among the following is an oncogene? It is c-MYC. Answer is option D. Identify the correct statement. Segmentation genes of Drosophila influence anterior-posterior axis formation. Segmentation gene controls the differentiation of the embryo into individual segments, not anterior-posterior axis formation. Anterior-posterior axis formation is influenced by bicoid and nanos. Hunchback is a homeotic gene. Hunchback, it regulates transcription of genes responsible for anterior structures. So it is not a homeotic gene. Hox is a maternal effect gene. Nanos, bicoid, hunchback, caudal are maternal effect genes, not Hox. So C is also not correct. Caudal is a maternal effect gene. Yes, that is correct. So the answer here is option D. The plant hormones involved in flowering is gibberellin. Answer is A. The antibiotic that suppresses prokaryotic replication. So the one which attacks the DNA replication mechanism, you have to find out. So streptomycin, it inhibits protein synthesis. Chloramphenicol also inhibits protein synthesis. Ampicillin, it interferes with the cell wall synthesis, and nalidixic acid inhibits DNA gyrase activity. So it is involved in prokaryotic suppression of prokaryotic replication. So the answer is option C. Now primase, it is an RNA polymerase. Primase is an enzyme that synthesizes short RNA sequences called primers, and these primers are starting point for DNA synthesis, and since primase produces RNA molecules, the enzyme is called an RNA polymerase. Then intercalating agent cause. Intercalating agents have the same size as that of the nucleotide. They produce mutation by intercalating between the bases of DNA, thereby disrupting or distorting the 3D structure of the helix, which result in single nucleotide insertions and deletions during replication. And these insertions and deletions, they produce frameshift mutation. So intercalating agent cause frameshift mutation. Answer is option C. Then next one. In E. coli, RNA primer for DNA replication is synthesized by E. coli RNA polymerase. Yes. E. coli replication is sensitive to rifampicin. Of course, because it is rifampicin act on RNA polymerase. So E. coli replication is sensitive to rifampicin. So both the statement are correct, and the statement 1 is the correct reason for statement 2. Answer is option A. A hypothetical gene of E. coli expresses to produce an mRNA of 120 nucleotide long. How many possible number of introns in this gene? E. coli doesn't have introns, so 0. Answer is C, 0. Which of the following statements about membrane carbohydrates is not true? Some are bound to protein, some are bound to lipids. Yes, glycoproteins, glycolipids. They show little diversity. No, they show diversity. Answer is option B. That is not true, they have asked. So B is correct. Yes, is the answer. They are added to proteins in the Golgi apparatus. Yes. They are important in cell surface recognition reactions. Yes. Then, in a human population, a rare X-linked single gene disorder has a frequency of 0.01% among males. What will be the percentage of affected females in the same population? So, in humans, females are homogametic, that is XX, and males are heterogametic, that is XY. Females have 2 X chromosomes, so for a recessive trait to be expressed in the female, both the chromosomes must be mutated. Whereas males, mutation of only single X chromosome is enough to cause the condition. So based on this concept, 2 inferences can be drawn, that is, since the male have only 1 X chromosome, the frequency of X-linked allele in the population is equal to the frequency of affected males. As females have 2 X chromosome, the probability of any female having the mutated allele in question on both the X chromosome will be Q square, where Q is the frequency of the allele. So as per the question, frequency of affected males is 0.01. So Q is 0.01. They have asked for frequency of affected females, that is Q square. So 0.01 square will be answer A. Answer is option A. Then which of the following is not a second messenger? ATP. Out of these, ATP is not a second messenger. Then linked genes. Linked genes are on the same chromosome. Yes, that itself is correct. Have alleles that assort independently? No. never show crossing over. Sometimes they show incomplete linkages, sometimes they show crossing over. Always have multiple alleles, no. Then a clade is a group of evolutionary related species that share a common ancestor. Answer is option B. Which among the following is not a pre-zygotic reproductive barrier? So what is pre-zygotic barrier? It is a mechanism that blocks reproduction from taking place by preventing fertilization. So it prevents fertilization, that is pre-zygotic reproductive barrier. So it can be through temporal separation, spatial separation, etc. All these are pre-zygotic reproductive barriers. Then post-zygotic barrier is a mechanism that blocks reproduction after fertilization and zygote formation. So you get a after the zygote formation only post-zygotic barrier happens. Before zygote formation, before fertilization, preventing fertilization is pre-zygotic reproductive barrier. So temporal segregation of breeding seasons is pre-zygotic. Hybrid infertility, hybrid you get only after a fertilization. So it is post-zygotic reproductive barrier. So the answer here is option B. Spatial segregation of mating sites, sperm that cannot penetrate an egg. Both these prevent fertilization. So the C and D are pre-zygotic reproductive barriers. Then predation and herbivory are examples of dash interaction. They are antagonistic. Antagonistic is one species benefits at the expense of the other. That is antagonistic. And here it is predation and herbivory. So it is option A is the correct answer. A group of individuals born at the same time frame are called. So they are called as a cohort. Answer is option C. Deme is any isolated subpopulation subjected to selection as a unit rather than as an individual. Subpopulation means you have got a population of sheep. Out of that you choose only very few a small population of this bigger population is called a subpopulation. Then taxon is defined as a collection of one or more population of organisms. Then next one, ecological succession is the change in. So it is the change in species over time. No, community composition after a disturbance. Yes, that is ecological succession. Then next one, which of the following is not currently a major cause of species extinction? Habitat destruction. Yes, it is. Rising sea levels. Yes, of course. Introduction of exotic predators. It is not a currently a major cause of species extinction. It is not. They have asked for current cause of species extinction. So introduction of exotic predators is the answer. Hollandric genes are Y-linked. The polyploidy condition which can be called amphidiploid. So amphidiploid is having diploid set of chromosomes from each of its parents. So if you see C, it has got AA from one parent, BB from another parent. So C is the answer. So it is having diploid set of chromosomes from each of its parents. Whereas in other option B doesn't come into picture. The next one. How many different types of male gametes are produced by a trihybrid? It is given by the formula. How many gametes are produced is 2 raised to n. Wherein here is trihybrid. So 3 are the number of individuals involved. 3 genes are involved. Or 3 heterozygous individuals are involved. A, capital A, small a, capital B, small b, capital C, small c. That is trihybrid. So here 3 genes A, B and C are involved. So 2 raised to 3, that is 8. So the answer here is option B. Which among the following is not true about a biopole? Biopole is the brand name of polyhydroxybutyrate, a biodegradable plastic. So it is biodegradable, yes. It is made from PHB, yes. PHB is polyhydroxybutyrate. It is soluble in water. It is not soluble in water but becomes swollen in water. Answer is C. Then next one. The approximate size of human genome is, it is 6.3 gigabases. Answer is option B. Which among the following is disadvantage of Bayesian algorithm compared to maximum likelihood? A specific mutation model is required. That itself is the answer. Answer is option A. Which among the following is not a domain interaction in multiple domain protein? So you have got a protein with multiple domains. So the domains between in the multiple domain will keep on interacting with each other. Now they have asked which among the following is not a domain interaction? So stable complex, yes. Non-stable interaction, no. It is not a non-stable interaction. It is a stable interaction. Transient interaction, yes. So answer is option B. Which among the following is not true for yeast 2 hybrid screen? A specific bait to prey interaction is assumed, yes. It is a method to study protein-protein interaction, yes. Non-specific bait-prey interaction do not affect the result. They affect the result. So answer is option C. The DBD is DNA binding domain and TAD is transcription activating domain are components of the yeast 2 hybrid system. Yes. Then which of the following is not patentable in India? Random DNA sequences, a plasmid vector, an artificial animal cell line, a cloning methodology. Random DNA sequences are not patentable in India. Answer is option A. State whether the two statements given below about plant variety protection in India are correct or wrong. In India, sui generis system of plant protection, plant patents are permitted. No. One is wrong. Farmers' right to their varieties are inherent in PPVFR Act of 2001. That is correct. So option D is the answer. 1 is wrong and 2 is correct. What is the full form of UPOV? It is International Union for the Protection of New Varieties of Plants. Answer is option B. The international agreement with specific depositions of microorganisms in IDA is Budapest Treaty. It is for microorganisms. A question based on Budapest Treaty was asked in July 2023 examination. I have uploaded the video. You can watch it there in my channel. Which among the following is patentable in India? A new design to an automobile. Answer is option D. State whether two statements about refinement in Indian patenting given below are correct or wrong. Diminaco AG case changed the concept of live organism patenting in India. Diminaco AG case was for patenting a live recombinant organism. 1 is correct, but 2 is not correct. So, it was a invention relating to a process for the preparation of bursitis vaccine, and this vaccine is useful in protecting poultry against bursitis infection. But the process to prepare the vaccine contained a living virus in the end product. So it was not been given patent. So he could not take a patent for this particular discovery. So statement 1 is correct, whereas 2 is wrong. Answer is option C. Then Geographical Indication Registry Office of India is situated at Chennai. A question on geographical indications was asked in July 2023 question paper. The maximum cell-to-cell junction is found in. So maximum cell-to-cell junction are found in epithelial cells. Gap junctions are absent in erythrocyte. Gap junctions are absent in motile cells as well as erythrocytes. So a question on gap junction was asked in July 2023. There I have told the same thing, that they are absent in motile cells but they are and also in erythrocytes. Then peroxisomes are produced from. They are produced from Golgi bodies. Answer is option A. The polysaccharide absent in plant cell walls is chitin. Chitin is present in the cell wall of fungi. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you are new to my channel, don't forget to subscribe. I have also uploaded previous year question papers of Kerala SET Biotechnology examination. You can go refer to my channel playlist for the same. Thank you for watching.